Chapter 1 is our introduction chapter. You might notice in this video that some of the topics differ slightly from what's in our textbook, and that's okay. You don't necessarily need to read all of Chapter 1 to go along with this. Definitely read Chapter 1.3, which is our section on the scientific method. There's also a video that will go along with that. And then you can sort of skim Chapter 1 and just see what is relevant. There's some of Section 1.1 is going to be relevant here. I largely skipped Section 1.2 because we're going to come back to evolution later and do several chapters on evolution. So I don't really introduce it right now. So as far as studying for Chapter 1, you are going to want to pay most attention to these learning objectives and not quite so much what's in the book. That's not to say the book is not important, because it is, but focus your studies on these topics. So we're going to talk about the role of model organisms in biology, in studying them in a scientific way to learn about the organisms on our planet, and why we use model organisms instead of always studying exactly what we want to learn about. This topic is not in the chapter, so you're going to have to get all the information from here. If you have questions, feel free to let me know. We're not going to go into a huge amount of detail here, but you do need to know what is provided in the lecture. We're going to talk about how to use the scientific method. There's going to be a video on this that you'll need to watch before we get to Lab 2. Normally I don't do that. Normally I don't have lab-specific videos, but this one goes along with our lab very, very closely. And that's going to be the last video lecture. It'll probably say Chapter 1, Part 3, which might be out of line with our other video numbering. Sorry about that, just a goof up. But anyway, going back to the learning objectives, we're going to create graphs of data sets and analyze trends within graphs. We're going to do that throughout the semester. I'm not going to talk about it too much specifically here, but we'll talk about it as it becomes relevant, mainly in our labs. We're going to talk about scientific theories. That is mainly going to be in with the scientific method video, although I'll touch on it in this first or second introductory video. And then we're going to talk about reductionist and system biology approaches to studying the world around us. Some of this is in our chapter, some of it is in the lecture. Now, normally our chapter lectures will follow very closely with the book chapters. This one just diverges a little bit for some reason. I don't know why. So let's get started talking about model organisms. So model organisms are organisms that biologists or other scientists use as a model system to learn about similar organisms. So many biological studies start in model organisms. So let's say we want to know what a particular gene does in humans. Well, it's not really politically or socially correct to experiment on humans. So if we really want to know what happens if a particular gene is disabled, we might do that in something like a mouse, something that's another mammal that is easy to study. And the reason that this works is that because of evolution, mammals are relatively closely related. So something that works in a mouse is probably going to be kind of similar in a human. Maybe not perfect, but it would at least give us a good idea. So using model organisms allows us to sort of focus our resources on a small number of animals that are really well studied. And I say animals, it can be many other things. It can be bacteria, it can be plants as well, fungus, things like that. But by using these model organisms, it allows us to sort of have sort of common research strategies, and it allows us to sort of focus on things that are related to what we want to study. And another reason for this is that sometimes you want to study something that is hard to get to. Maybe you're trying to study an endangered animal and there's not a lot, so you don't really want to be experimenting on them. Or maybe you're trying to study something that you don't have a lot of access to. Maybe there are not these animals in zoos or whatever it is. Or maybe you're studying some sort of crop and you want to make a change. So you might do it in something that's really well studied before you start getting to some new crazy experimental thing. So there's lots of reasons that we might use model organisms. Some of the more specific reasons for using model organisms is really that similar organisms behave in a similar way. Now, I just mentioned this, and this is because of evolution. So a lot of our course is going to focus around the concept of evolution. Now, the reason that evolution is important here is that organisms who are closely related are going to behave in a similar way. And what I mean by this is that similar processes in related organisms are going to be similar. So, for example, blood in pretty much all animals is really similar. So if you study it in one organism, there's a really good chance that those properties are going to apply to other organisms. Now, if you get down to some more specific animals, say something like a lizard or a mouse, you might choose to study a mouse if you are thinking about humans, because mice are more closely related to humans because we're all mammals. So because we're mammals, our systems are going to be more similar to one another than we are, say, to a lizard or maybe a chicken. And the reason for this, related to evolution, is that DNA is a universal genetic code. So we're going to spend a lot of time talking about DNA and how DNA works, how it actually does things in our body. But the important thing about DNA is that all living organisms have the same genetic code, meaning the same few letters, A, C, T, and G, are the language of all the genetic codes in all living organisms. And so the consequence of this, or the result of this, is that gene function is universal. So a similar gene in a related organism is going to have a similar function in both of those related organisms. And even going way, way back to early evolution, where all living organisms were single-celled, they were basically bacteria. There are many genes that are very similar, even today, between bacteria and, say, humans. Now, they're not perfect, but when you start looking at sequences in gene function, when the sequence is similar, they tend to have a very similar function. And so using model organisms allows us to sort of go back to the most basic, easy to study organisms and then apply that information to more complex ones. I also mentioned that it's more ethical to study animals than humans. Now, I'm not saying that there's not ethical questions about studying animals, but at some point you have to have an alternative to studying humans. And because I think everyone is going to agree that it's not ethical to experiment on one another. So by using model organisms, we can avoid experimenting on people. Another reason to use model organisms is that they're cheaper to study. It's cheaper to study animals. It's cheaper to study small animals. It's cheaper to study small plants. And that idea of being sort of cheaper and easier to study model organisms relates to them being easier to grow, easier to house, easier to care for. If you're caring for a colony of mice, it's going to be far cheaper than caring for a colony of humans, right? So mice are also small. Fruit flies, another common model organism, are very small. And food is cheap for them. They're easy to feed. They're easy to care for. Another advantage of model organisms, particularly if you're looking at genetics studies, is that they have relatively short life cycles. Now, this means that you can study many generations of these model organisms in a very short period of time. So you're looking at several months for generations rather than decades with people. And related to that, they also tend to produce many offspring. So especially when you're studying genetics, statistics is key. So if you look at one organism and say 2 of its offspring, you're not really getting a good idea of if whatever characteristic or problem you're studying happens to everybody. But if you're looking at fruit flies and you have hundreds of offspring, you can get a better idea of what's actually happening. But unrelated to genetics, having short life cycles and many offspring gives you a lot of options for breeding, for looking for trends, and things like that. If you are talking about genetic analysis, using model organisms is good because they're going to have less genetic material. And so that just tends to make genetic and molecular studies easier when you're dealing with less genetic material. I'm not going to go into what exactly that means. Just keep in mind that that's kind of important when we're looking at model organisms. And then we have less complex systems in model organisms. Now, you might look at that mouse and be like, well, it's got a heart, it's got liver, it's got kidneys, it's got blood. They do, absolutely. So not all model organisms are going to be less complex, but they are going to be really well studied. So all of the systems in the mouse or the fruit fly are going to be so well studied that you can find information on anything that you want to study, and that's going to help you with your particular research problem. And I'll talk about some other organisms in a few minutes, but there are actually model organisms who are less complicated, who have less complicated nervous systems, who have less complicated development, less complicated bone structure. And all of those things make model organisms much easier to study than higher order complicated organisms. So a couple of examples of model organisms. The first one, probably the most common model organism, but probably least known to you, is Escherichia coli, or E. coli. Now, E. coli is important because it's a bacterium, which means that it is single-celled. So this guy right here is one entire organism and just one cell. These little dots on this plate here are a colony of them. So you might have millions of bacteria on that plate that you can study. Now, the reason that we want to use single-celled organisms as much as possible is that they're far less complicated. And remember I said that DNA is universal. The genetic code is universal. So if you're looking at really simple genetic processes, genes, things like that, you can start with bacteria and say, hey, what do they do in bacteria? Because if that gene is present in something more complicated like humans, then you might get an idea of what that gene does in a human. Now, this begins to break down because E. coli are far less complicated than humans, so not everything that we have, they're going to have. But it does give a good starting place if you're looking for something that might be universal. E. coli is easy to obtain. Now you just purchase whatever strain you need from a biological supply company. You can even produce your own types of E. coli if you need to. And you might be thinking, oh, E. coli, that's the thing that makes you sick. Absolutely. There are strains of E. coli that do make people sick. This is the one that tends to, if you get food poisoning, it's usually E. coli. There's always recalls for things like lettuce or meat that are contaminated with E. coli. But the reason they were used originally is that they're found in your gut. So they're found in your intestines, and many people have E. coli in their intestines that are not harming you whatsoever. Most of these E. coli strains that are occurring naturally are actually benefiting you. They're helping break down things in your intestines. There's a whole vast colony of bacteria living in your gut that are doing really important things for you, like helping you break down your food and providing vitamins for you. But they were originally obtained from stool samples. They're easy to get when there's no biological supply company and you need to study something. E. coli is important because it helps us understand complex molecular processes in a really simple organism. And then now there are lots of mutants, knockouts. These are knockouts are organisms where a gene has been disabled, so you can study what function was lost when you took out that gene. And then E. coli, like all other living organisms, are susceptible to viruses, so they can help you study different types of viruses. Specific viruses will infect E. coli, and so early studies on viruses were actually done in bacteria. Another common model organism is Drosophila melanogaster. This is more commonly known as, as the fruit fly. So this is the guy that flies around your kitchen and infests peaches and things on your countertop in the summer. Same little guys. And they are important because they are one of our eukaryotic or multiple-celled organisms that are model organisms. They're an insect. Obviously, they're a fruit fly. They meet several of our criteria for model organisms. They have a short generation time and lots of offspring. That's why you can get an infestation of them in your kitchen really easily. The fruit fly was used in early genetic and chromosome studies, again, because they have very simple genetic makeup. They have only 8 chromosomes, whereas we have 46. So doing genetics experiments with them is much easier. Just like E. coli, there are many different mutants. There's some examples here. We've got some color mutants. We've got eye color mutants. There are wing mutants, all kinds of things. And so you can study developmental processes. You can study gene knockouts here very easily with Drosophila melanogaster. One of the cool things about them is that they were one of the first organisms to have their genome entirely sequenced as early as 2000. So that allows for genetic comparison of sequences. Because again, as I said a few minutes ago, a similar sequence is going to provide a similar function. So that can help you understand what the gene you're looking for is doing in the organism you want to study. Another common one is the mouse. This is just sort of a common laboratory mouse, these little cute guys with pink ears and pink noses. They're really important because they're a mammal. So you can study a lot of diseases and gene functions in mice that are going to have really similar outcomes in humans. And so they're really important in medical studies. They have high homology with the human genome. Homology means same. So the human genome, or the sequence, the code of our DNA, is really similar to the gene sequence in mice. So some genes are up to 99% identical. Basically, they're the same genes. Some are only 60% identical. That's because of evolution, because those genes have been put under different pressures over evolutionary history of both humans and mice. But because they're still relatively close, we can still use some of those genes to sort of figure out a purpose in humans or figure out diseases in humans, because something that affects an organ in a mouse is probably going to affect it the same way in humans. Or you might start drug development in mice. What does this particular potential drug do to mice? Does it affect the disease in the way that we want it? And then you can apply that to humans later through the right pathways of study to see if, in fact, it's a viable option. Just like our other ones, we have lots of mutant strains. We have transgenic strains. Transgenic means they have genes from other organisms to do other things. There's a couple examples here. This one actually has a fluorescent jellyfish gene that has been applied to places that are expressed in the ears, nose, and mouth. And tail. So that looks really cool, right? That's actually, there's a scientific purpose for that a researcher wants to see where this particular gene is being expressed. But obviously we can't do that to people. It'd be really cool, but we can't do that to people. This one over here, this is a mouse that has a defective gene that is important in obesity. Obviously, you can see that this mouse is much larger than the normal one over there. So another way that we can use the mouse for studying scientific anomalies in humans. So there are a few other model organisms. Each one has its advantages and purposes. Sorry, there's some typos on this slide. There's a couple here. Nematodes, they're this aquatic worm. They're really cool because they're clear, so you can see all the way through them. You can see the development of their different organs. There's been a lot of neurological studies in here. In fact, the development of all of their neurons is actually mapped. This fish here, the zebrafish, he's really cool because his babies are clear and they're small, they're easy to study. So there have been a lot of development and neurological studies on them as well. Getting away from animals, we have yeast. This is like baker's yeast. The reason that people started studying this is that it was available because people were using it in their bakeries, so you could get it. And so that is a single-celled eukaryotic organism. So we'll get into prokaryotes and eukaryotes a little bit later, but it's basically a really simple organism that has the same type of cells that humans and other animals have. And then, of course, we need a plant because plants are important too. When you have a small plant, this helps you basically in the same way that our mice or fruit flies help studying people. When you have a less complicated plant, you can more easily study the functions of genes and disease and things in this little plant that's easy to grow. So there's a few other examples of human diseases that can be studied using these different organisms. I'm not going to go through all of them. You can kind of look through and see if there's anything that interests you there. You don't have to memorize those diseases, just sort of understand the purpose of how these model organisms are used.